Now, uh, let's move to the agenda. Today, let us try to discuss about the proteins, their structure, function, and importance. And next, I will move to the topic of today's discussion, which is enzymes or proteins for industrial application. And then we will move to see what is a protein engineer, what is protein engineering, and then why do we need to engineer protein. So for this, we will look into the protein engineering concept, the methods, and I will take you through a case study, and then we will discuss about the challenges and what is it that's happening in the Indian scenario. And finally, we will move to the summary and way forward. So when we talk about proteins, of course, I'm not, trying to educate you on the structure function of proteins, but I'm just trying to reiterate what proteins are, what do they do. So as we all are aware, proteins are large, complex molecules, which are very critical to the normal functioning of the cells or the muscles, tissues, whatever you call. The proteins form about 20% of the human body, and they are made up of amino acids, as you can see here, which are joined together by the peptide bonds. And proteins, of course, they have different kinds of or different levels of structure. When you have the amino acids joined together by peptide bonds, this forms the primary structure of the protein. And then we have alpha helices and beta pleated sheets, which are referred to as the secondary structure of the protein. Then we have polypeptides forming together, or just the polypeptide chains. At this level, you can call them as the tertiary structure. And further arrangement of these polypeptide chains is referred to as the quaternary structure. And what is important to remember here is proteins are indispensable and they perform various activities in our body. So if you look at the the schematic or diagram that I have presented here, they act as enzymes. Say if, the entire gut system of our human being is having a lot of these enzymes. We have amylases, lipases, pepsin, trypsin, all those coming into picture, which helps in the digestion of food that we take in. Hormones like insulin and thyroxine, they are also of protein origin. Similarly, we have the transport function performed by protein. So basically, we are talking about hemoglobin here. And for the structural integrity of the cells, proteins are very much required. Example, actin, tubulin, keratin. These things come into picture. It's not just here. We have proteins in the defense system acting in the contraction and relaxation of muscles, and even as storage proteins. So this is about very briefly the function of protein in our body. And then we will move to what exactly is, what exactly are enzymes? Why are we talking about, protein engineering? So of all these, if you look at the first panel here, the enzymes play a very important role. It is not just in the digestion, even several or all the metabolic pathways in our body are mediated by these enzymes, or many enzymes for that matter. And enzymes are the most important class of proteins for industrial purposes. So that's why today I'm discussing about what these enzymes are, how are they used, and what are the ways in which we can have industrial enzymes. So when I say or when I talk about enzymes, so we have various enzymes that are actively being used in the industry. So I was closely associated with the enzymes for the detergent industry and enzymes for baking and enzymes for even several industrial processes. And if I have to just draw your attention and just to create some curiosity, most of your detergent that you get in the market today, especially the powder and the liquid, so they do have amylase, protease, and lipase. And just to give you an example, be it like Surf Excel, whatever is supplied by Unilever or many big players, you will see some colored structures present in the detergent powders. Basically, they are the formulated enzymes that help in the degradation of the dirt or dust, whatever you call. What basically happens, these enzymes, they act on the carbohydrates, the lipids, and the proteins, that are sitting on the cloth. And once these macromolecules sit, they tend to attract even further dirt or dust or whatever. And that creates the stain. Basically, these amylase, protease, and lipase, they break down these complex molecules into simpler or short chain molecules. And then these short chain molecules, they get lifted off from the stain, and it's easy for the associated molecules also to come into the wash liquor, right? Similarly, we have baking industry that makes use of amylase, protease, lipase. I'll not go to the details of it, but the dairy industry, the pharma, uh, food and feed industry uses it. Biofuel is one thing, one of the projects which I worked during my postdoc studies, which makes use of glucoamylase. So enzymes are also extensively being used in the textiles, leather industry, beverages, paper and pulp, and agriculture, and finally, the waste management. So when I'm talking about this, now you have already realized that I am talking about different environments. If you look at the detergents, the environment that the detergent has is different. And the baking industry operates at high temperature in some regions or some parts of their workflow. Then we have dairy. All these, enzymes that are present naturally are not, ready to work in these environments. And if you look at the business, the global enzyme market is very huge. And this was estimated, I have the data from 2023. So it was estimated at uh, 60 billion US dollars in 2023. This speaks for the kind of activities that are associated with industrial enzymes. And all these, why is this happening? The countries which are like, the developing countries, developed countries, all of us are now talking about the green chemistry. All of us are talking about sustainable development. And the best answer for this sustainable development is enzyme mediated industrial processes. So that's why most of the industries are very slowly moving towards this sustainable growth. And as I just mentioned, the enzymes need to work in different environments. And this is possible only because these enzymes were engineered, or this is because of the protein engineering. Now comes the question, what exactly is protein engineering? Is it that these enzymes are not able to work? If not, what is the reason for these enzymes, not active at these different environments? Now, if you look at a simple, answer like this, can natural, enzymes work in different conditions? The answer is no. So basically, as I just mentioned, the industrial applications have different harsh environments. Say, for example, there are many industrial processes that happen at low or high pH, and sometimes very high or very low temperatures are required for the industrial processes. Some of the processes, I'm talking about different industries, it's not just single industry. Enzymes may need to work in low water conditions, or it might have to work in environment that has surfactants. The best example is the detergent. So, and sometimes enzymes need to work in a very long process of 6 to 7 hours. And other denaturants are also present in the environment where the enzymes should work. That's why these enzymes need to be engineered depending on the industrial requirements or the applications. Now, what do we have? Or, what exactly is protein engineering? And then why do we have to engineer? There are plenty of examples that already exist in the literature, but I'll not be talking about those examples. As I just mentioned in my previous slide, you could think of starting from dairy to waste management to biofuels, these enzymes are being used. And these enzymes are all engineered. And now to define the protein engineering or enzyme engineering, to be precise, it is just about, it's a biotechnological, technique that aims at producing new enzymes or proteins, or it aims at the enhancement of capabilities of existing ones. So either you find or make a new enzyme or increase the capabilities of the existing enzyme. And scientists employ these protein engineering techniques to design and create proteins with useful properties. As I mentioned earlier, the proteins are not meant for working in such harsh environments. Wherever the enzyme is present, it is suitable for working at that particular environment. Say, for example, you have bacteria that live in cold environment, and the enzymes that are present in these bacteria, they are adjusted, or their amino acid sequences are evolved in such a way that the protein functions best in the cold environment. So there are various aspects of protein engineering, and then why we use protein engineering. And these are some of the examples. The pH and temperature stability. If you want to work at low pH, high or low temperature, you need to stabilize the enzyme for that particular environment. Similarly, some of the enzymes are inhibited by substrate product. You have various kinds of inhibition, and increasing the substrate affinity is one method which increases the activity. It's not always true, but there are many examples like that. And this is one important aspect where industry looks for protease resistant enzymes. And these enzymes can also be made with the help of the protein engineering. And we have active enzymes in the non-aqueous solvents. There are some reactions that occur in less water conditions or non-aqueous conditions. Even we can engineer the protein or the enzyme using the engineering methods. Stability to detergent is what I just discussed about. And sometimes you can make the enzyme free from its dependence on the metals or cofactors. And if you want to have novel functionality, that means whatever is not existing today, that can also be done with the help of protein engineering. Now we have understood simply the methods, not just the methods, sorry, we have understood the use of protein engineering and the potential of protein engineering. Now, coming on to the methods and principles. And if I just give you an example of these two pictures, you will understand what we are going to do. And look at these pictures. A person is, using this parachute. This is in the air, and a person is using the oxygen cylinders in the aquatic environment. So this simply means we need different tools for different environment. And the same principle holds true even in protein engineering, where we need different kinds of amino acid sequences in protein to tolerate different environments or to work in different environments. Simply put together, the principle of protein engineering is that by modifying amino acid sequence of a particular protein or enzyme, you can change the structure, function of that particular enzyme or protein. This sounds interesting, but it has many challenges. But how do you identify the mutations or the changes that you have made? So it is through the development of assays or high throughput screening. And once you change the amino acid sequence of a protein or an enzyme, you should basically have some assay to identify the impact of that mutation on the protein. So by developing suitable assay, you can actually select the proteins that have additional capacities compared to its original backbone or the wild type. Now, let us get into a hypothetical condition or situation. Basically, I have selected this to make us or to understand how a typical protein engineering works, right? So I'm not revealing any of the methods or techniques or tools or whatever that I did in my previous company, since it is agreemented or whatever you call patented, it's very much available. But through the literature and also with my experience, whatever I have gained, I would like to take all of us through a journey where there is a requirement from the industry. They are looking for a thermostable alpha amylase that can act at 70 degrees for 6 hours. And even after this, it should retain 80% of its activity. So this looks like a Herculean task because most of the enzymes that we have seen, they work at 38 degrees or 40 degrees, and it might continue to, deliver its activity for, let's say, 1 hour or 2 hours or so. But the industry is looking for an alpha amylase. So alpha amylase is basically the amylase that breaks the starch and glycogen into maltodextrins and short chain oligosaccharides. So, but the industry has come up now with this requirement where they want activity for 6 hours, like 80% activity at 70 degrees. Now, the typical question that a protein engineer asks or should ask is, how do I select the organism from which I get my enzymes for engineering? So if you are lucky, you can directly get some enzyme that act at 70 degrees for 6 hours, 80%. There are cases. But if it is not the case, then you have to first identify an organism from which you have to, or from which you are planning to get the amylase. Then the next part is, if the amylase is not delivering whatever I need, then you have to modify the amino acids. You have to, start this protein engineering process. And what are the methods that is available is what you need to think of. Then finally, I have selected my enzyme. Let's say, for example, I have mutated the protein. I'm seeing some activity, but how do I select the better protein from screening? So these three basic questions should be asked by any protein engineer. So assays are quite important. And unfortunately, even the best of the best reviews I went through, they have failed to recognize the importance of the screening methods. That's the biggest bottleneck. And of course, I don't want to comment on the research that has been made. But yeah, now look into this and let's wear the cap of a typical protein engineer. Now, the industry has come to us. They're asking for an enzyme that has or that can perform at 70 degrees for 6 hours. Even after 6 hours, it should retain 80% residual activity. Now, where are these enzymes, especially these alpha amylases, are used? As you can see here, I have just represented the action of amylase. The polysaccharides like starch is acted upon by amylase, alpha amylase. It breaks the starch into oligosaccharides and disaccharides. And these are very much used. Amylases are used in detergent, food, textile, pharma, and bakery industries. The person has now come with the requirement that 70 degrees, 6 hours, 80% activity. Now, how do we go about it? How do we produce this enzyme? Now, the first question is the selection of organism from which I can get this enzyme. Now, you would have realized that the requirement is for an enzyme, alpha amylase, which should be active at very high temperature. Obviously, you will think of the organism which grows in very high temperature. The logic or the principle is that those which grow in very high temperature would also have enzymes working at high temperature. And the options that you basically get is the psychrophilic organisms, that is, which live in cold environments, typically below 15 degrees centigrades. And the second option that you get is the mesophilic organism, just, which live in temperature between 20 to 45 degrees. And you also have thermophilic organism, which live at very high temperatures. Some of the bacteria and then archaea, which are found in the hot springs, they are available to you. And ultimately, hyperthermophilic organism. So just to take a step back, the industry has come to us. They have requested for a thermostable amylase. Now we are doing a brainstorming session and we are looking at three steps. The first step is, where do I get my amylase gene? For that, there are different environments that we are talking about, starting from cold to the hottest environment. Now, the answer is simple. You can go with the thermophilic organism because till 80 degrees centigrade, centigrade, these enzymes can act, or lesser than that. And our requirement is somewhere falling around 70 degrees. But at the same time, the catch here is these thermophilic proteins or thermophilic enzymes are, they are rigid. Rigid in the sense that they have structures in such a way that the unfolding of protein is resisted, under extreme harsh conditions. So this also means that their activity is sometimes, compromised. That's why people usually go for mesophilic organisms where you have good activity, but the stability is missing. Now, the next part is you have selected the organism, either mesophilic or thermophilic is what you are trying to select. And what are the enzyme engineering techniques that are available today? There are many, many techniques. And for the sake of understanding, I have, you know, grouped these enzyme engineering techniques into four major ones. So you will also see a lot of PCR techniques, how to generate mutant proteins, how to generate, DNA shuffled ones, all those stuffs are there. Let's not get into the deeper aspects of it. So we are just interested in producing mutation in this protein. And after that, we will screen or we will test whether this enzyme is meeting our target. So for that, you have basically four different types of protein engineering or enzyme engineering methods. As the name itself indicate, the first one is directed evolution. So where, very recently, I don't remember the year, Frances Arnold, she got the Nobel for this, where you are directing the evolution of protein. That means you are changing the amino acid sequence of the protein, and it is not in a particular location. You are not targeting a particular amino acid, but you are randomly mutating the gene. And as a result of this, you will have proteins carrying different, amino acid sequences. And sometimes it is desired, sometimes it's not desired. What you are trying to do here is you are simulating natural selection, but you are artificially introducing these mutations. Then, by using different screening methods, you will identify proteins that are better than the wild type. The next one is the rational design. So there is a rationale for this kind of design. It's a classical method which I have extensively used. And this basically looks at site-directed mutagenesis. Instead of, mutating at different regions of protein, it actually, with the help of literature, with the help of knowledge that is pre-existing, we basically tend to decide on the amino acid to be mutated. So specific point mutations are made based on the knowledge that we have gained. And after that, a robust screening method is established, and we will identify the ability of this enzyme to perform at higher temperature. Okay. This is technically easy, relatively inexpensive, but it requires prior knowledge of the protein. And then comes a combination of both. That is why we call this as semi-rational design, where we combine the methods and technologies used in directed evolution and rational design. And we ultimately, in all the methods, we develop proteins with different amino acid sequence, and we will check whether it has additional properties. Finally, David Baker, who got the Nobel Prize last year, if I remember correctly, that is for de novo design of protein, where this is completely different from what you have heard so far. This aims basically at producing proteins with particular topologies, with different folds, with different catalytic sites, and sometimes it generates new proteins with sequences which is not present in nature at all. Right? Now, the second part is what we have concluded. First is we have zeroed in or boiled down to mesophilic organism or thermophilic organism. We have decided to isolate the amylase from them. And now we have looked at the enzyme engineering methods. And without going into the details of this, I would say that let's just concentrate on rational design and semi-rational design. Now we know the methods. What is it that we are going to do now? I have given you, this is also available in the literature, not that I'm just talking about my work, but it is indirectly related with that also. Now, coming to the enzyme engineering part, the case study, I have also mentioned earlier that we are looking for an enzyme amylase that acts at 70 degrees for 6 hours. Even after that, it should be retaining 80% residual activity. Now, how do we do that? It's theoretically simple. What we basically need to do is select an amylase donor. That's what we need to do. That is the amylase, you know, if you want to take it from thermophile, if you want to take it from mesophile, decide on that. Then you clone, and after cloning that amylase gene, you transform this to a protein expression system, or once you have this expression, check after purification, and whatever is the stability, you report that, and then see where are you or how far you are from the target. So I have given you one quick simple example of what is done here. And as I just mentioned, you have selected the donor now, and from the donor, using different molecular biology tools or techniques, you will have to identify, sorry, you will have to isolate the amylase gene, and now your gene is ready, whatever you are seeing here. And now you have the bacterial expression systems, and in the plasmid that you have in, let's say, bacillus system or E. coli system, whatever you want to take, you create nicks in this plasmid, and then just join. I'm just trying to be uh, you know I'm just trying to explain this in as layman's language as possible, as I don't want to go to the details of it. Now you have the plasmid and you have the gene of our interest, and from whatever we have decided upon, that is from the mesophilic or the thermophilic environment, now transform this to a bacillus expression system. Now the plasmid has uh, its own genes. Along with that, we have amylase. And what we basically do is these, amylase carrying bacillus is grown or cultured, and the protein that is secreted out is purified. And now, request you to pay some attention here. Now the question is, the industry is asking for thermostable protein. And first, we need to check, is it really active? Is it really thermostable? And if yes, to what extent? And for that, what we do is the protein, that is, the enzyme that is expressed, amylase, is taken. It is incubated at 37 degrees to start with, I'm telling, for 30 minutes. And once we have this incubation completed, we take out the sample containing alpha amylase, and we check the activity after incubation. And as we know, amylase acts on starch. For the sample that is incubated, that has already undergone heat stress, we will give some starch, and we will see how, to what extent it can release the oligosaccharides. And the reaction that you can see, and I have just taken PABA assay, which is a reducing and measurement assay, as an example here. So alpha amylase that we have incubated now is acting on the starch, and it is breaking down into oligosaccharides and disaccharides. The PABA assay detects it and gives a yellow colored complex. And is it really active now? So we have selected the enzyme, we have put it into bacterial expression system, and then we are checking the activity. To what extent is it active? And if you look at this graph, we have tried to measure the activity of incubated protein sample. The one in red that you see here is the regular enzyme, which is not incubated. So you are just checking the amylase activity, and you will see that it is 100% active. And at around 38 degrees, you will see its good activity. So when I say activity, it still converts the starch into oligosaccharides. It's still active. And as we move down, in the sense, as we move along this temperature gradient, at 60 degrees or so, you will see that the enzyme is completely dead. The incubated enzyme, the second one, the second graph that you're seeing here, is already dead. Dead in the sense, I would say it retains about 65% or 70% activity after incubation. That means it's a scary situation now where the requirement is 70 degrees for 6 hours, but my enzyme is active for just 30 minutes at 37 degrees centigrade. This is what I say set the target. Now determine the half-life of this protein, that is amylase here, and then also finally you just calculate the half-life that is required for reaching this condition. Now it's a huge Herculean task that you need to start. So how do we do that? Now the answer is simple. We need to mutate the protein, and we need to mutate. After mutation, we need to identify the good proteins. There are many ways. As I just said, uh, there are many methods by which you can identify the regions of the protein that are unstable. And what basically happens at higher temperature is the amino acids which are interacting with their surroundings, they tend to lose these connections. The loops usually open and the sheets tend to spread apart, and you will see unfolding of protein. Now, what we basically need to do is we should look for the mutations that will help in stopping of this unfolding at high temperature. And there are many inspirations that you can get from. One is search the literature for mutation that induces stability. And the other method is we tend to align the sequences of the amylase that we have got with that of a thermophilic amylase. And when you align the gene sequences, you will see in some regions the consensus sequence, the sequences that are matching. And in some areas, you do not see that. So that means the thermophilic protein is different from the regular one. So identify these regions and then select for mutation. And also from the thermophiles, you have some inspiration where usually thermophiles will have a lot of prolines, especially in the loop regions. And they also have lesser methionine. Similarly, there are many concepts that you can look into, and a couple of other examples are you should basically target the surface exposed residues which directly come in contact with the environment, which is hot temperature here. Mutate them and then stabilize the metal binding sites. So once your protein starts unfolding, there are chances that the metal ions which are present often in the catalytic site, they tend to get leached out. So you stabilize those binding sites. I can just go on listing because if you look at the literature and published patents, there are many methods by which you can stabilize the protein. When you're looking into increasing the activity of protein, specific activity, then you would follow a different protein engineering technique. So here, what you are trying to do is you're trying to stabilize your protein at higher temperature. That means you are trying to avoid the unfolding of protein at higher temperature, and you are trying to stabilize different parts of the protein. And the analysis of crystal structure would also give you a lot of information on which region of the protein to target. Now, for my example, I have selected this wild type alpha amylase. As you can see, it has A, B, and then C domains, which sometimes they call CBM or carbohydrate binding module. And I'm talking about now rational design, where the gene of interest is mutated at different regions. And why are we mutating at these regions? We have got the inspiration from the literature, from the crystal structure, and also some computational study that these regions might give stability to protein. So that's why we are trying to change the red dotted regions of this gene. And for example, I have selected 6 regions of the protein. As you can see on the top, you have the wild type alpha amylase that we have taken. Now I am mutating that in the region number 1. So you can call this as mutation number 1. And the second region, which is again loop, and then you can call this as mutation 2. The third one, mutation 3. the fourth in the C domain, and similarly, fifth and sixth. Now, we have generated the mutants. What basically we need to do is, of course, these after the mutation, you need to transform into an expression system, and you have to express amylase in the expression system. Now that part is done. We have selected the mutations in different regions of the protein, and then we have transformed this into different bacterial system. We have expressed and isolated the protein. Now do the same thing that you did before, where you take a control and then you take the protein, that is, amylase, in different test tubes. The numbers here, 1, 2, 3, 4, 5, 6, refer to the enzyme carrying mutation at this particular region. So what you do is you take the protein, you incubate in the same temperature that you did before, 37 degrees for 30 minutes. And what happens after that? You better residual activity or activity even after this incubation, you select that. So by this method, let's assume that we have got the highest activity in the enzyme carrying mutation number 5. And let's say mutation number 3, mutation number 6, and mutation number 1 are followed. And finally, what you need to do is you have to select a variant, that is the mutant carrying mutation 5 as the backbone or the mother molecule for the next round. So initially, wild type was the backbone or the mother molecule for us, on which we introduced different mutations. And we identified that mutation at region number 5 or in this loop gave better activity. So just for the sake of understanding, I have given this. Okay, we are testing the activity at different temperatures, and then you will see that wild type retains about 20% residual activity at 40 degree or 45, whereas the mutation 6 retains 38% residual activity, and the 3 and 5 respectively retain 50 and 65%. So this is just for the sake of understanding. Do not start judging the lines or axis here. So the simple quick message is, after incubating these variants or mutants that you have generated, you would measure the activity, and then you will end up with identifying the mutations that give additional stability. And now you have identified a fifth mutation to be beneficial, and you have selected this fifth mutation as the backbone for your next round. Now comes the question, what should I do? My fifth mutation carrying protein or the amylase is good, but what should I do? In the next round, we are supposed to take this as the backbone and add the other mutations which were good, namely the mutation 3, mutation 6, and mutation 1. And now these are called double mutants. So these are carrying 2 mutations. And when you check, again, you repeat the same process. You take a control, and to that you compare the residual activity of the double mutants carrying 5 and 3, 5 and 6, and 5 and 1. Again, you will check the activity, and you will recognize, let's say you recognize that mutations carrying 5 and 6 gives you the highest activity, right? And this is also a very theoretical diagram that I have written. Wild type was retaining around some 10%, and mutations like 5 and 6, they are retaining 40% residual activity. And you will not stop here, and you will now take up the variant or the double mutant carrying 5 and 6 mutation, because this was the best in this round. And this acts as your backbone or the mother molecule for the generation of variants in the next round, which is the round number 3. Now, we have selected the variant carrying 5 and 6 mutation because that gave better residual activity. And in this round number 4, you will add additional mutation, which you would have identified from various other methods, right? It is not just the 5 or 6 that you start with. Example is what I gave. You would have started with some 15, 16 mutation. Now, those hit, we call that as hit, right? Those stabilizing mutations you start introducing. And again, in the round 5 and round 6, round 10, whatever, it continues till you achieve your target. Now, for the sake of understanding, I have given here that the ultimate molecule finally, or the final variant with the desired properties carries 15 mutations at various regions of the protein or various region of this amylase molecule. Now, we have achieved a Herculean task. We have solved it. Initially, the wild type had stability or the activity of 70% at 37 degree for 30 minutes. And now we have come up with a variant or a mutant protein carrying 15 mutations. And this is able to tolerate 70 degrees. It's able to act for 6 hours, and it is able to retain 80% residual activity. Now, any enzyme, any protein for that matter, can be, mutated, changed, but there are many challenges in protein engineering. One of the things that I just mentioned, which I try to, include here, is the screening and testing. We are not just talking about 5 or 6 mutation when you go to the industry level. We are talking about 5,000 to 6,000 different mutants, and only then you will be able to. Sometimes, if you are lucky, even 2, 3 mutations give you that molecule. But yeah, let's move on to the major one, screening and testing. Developing a robust assay is the biggest challenge that we have today because the high throughput screening comes into picture where you miniaturize the assay that you have developed, and the assay itself will have a lot of background noise. Sometimes you get false positives, false negatives, and there is evaporation issue when you work with the high throughput screening plates. There are many things associated with it. Unfortunately, the reviews that I went through, they're not stressing upon the the role of the screening and testing. This is one of the bottlenecks which the scientist is facing today. And there are various AI/ML stuff that has come into picture. Just like you have large language models, you have protein language models coming into picture. Deep learnings will also help you in identifying specific regions. And the tools like AlphaFold can tell you, even if you do not have the protein structure. That was the biggest bottleneck which we had. But now that's being solved by the latest technologies. And in addition to that, I don't want to just read this. We have de novo designing that comes into picture, but predicting the proper protein folding is not so easy. Predicting the conformational change is also not easy because whatever you do, even the simulation, if you are talking about MDMM simulations or whatever, you are creating an artificial environment. But when the enzyme goes to a buffer system and when it binds to the substrate, the properties are completely different. And when it starts releasing the end product, its stability, uh, if you talk about the stability level, it is different. So that's why the challenges, especially predicting these transition states, predicting the conformational changes that happen before and after binding of substrate, before and after the reaction is completed, and then finally, its rearrangement into a catalytically active condition. So these are some of the challenges that we have, but AI is, solving many of these. But I'm yet to hear some good successful stories. There are, of course, but some concrete evidence should be there. Now, coming on to the very reason for me selecting this is, just to get your thoughts on what's happening in the Indian scenario, because universities, they definitely have a lot of potential, and the companies which I'm talking about, the enzyme industries, We hardly have few names that I have mentioned here. Advanced enzymes, Infinita Biotech, Enzyme Bio, and then in Biogen Technologies. But so far as my knowledge is concerned, we are still far away from the multinational companies in creating the enzymes which are required for the industries. What basically we are doing in India is either getting these enzymes from the big companies, reformulating, adding some other company enzymes, stabilizing it, diluting it, and then selling it to the customers. So it would be really good if the Indian universities and professors and researchers come forward and then explore the biodiversity, because we have the kind of variations that you get starting from cold environment to the hottest environment. It's very, very difficult to get in any part of the world. You might get, but the kind of work that we do, the kind of diversity that we have is unparalleled. If somebody could think of it, definitely it would be a good idea. And even if it is given as a project to the summer students or whatever, collection, categorization of bacteria, isolation of some enzyme, which is industrially important. Now, a lot of industries are moving even for the drug discovery or whatever, not drug discovery, sorry. So synthesis part, they are looking into these enzymes, right? And I can recall one human genome project where, one of the big organization, in India, they made use of the students who were visiting there as interns. From different states, they could collect the human samples. Why don't we do that? Right? That could be a better thing. Even if you collaborate with industries that are producing these, or at least the big players in the market, and then offer to them that I have so much of diversity sitting in my laboratory. Can we explore together? It can generate huge funds. I don't know to what extent it can be sold to the other um, industries, countries, but I would request the community sitting out here to look into this. And yeah, to summarize, enzymes are futures, if you ask me, with my personal experience. And most of the industrial processes are now being, you know, replaced by the enzymatic methods, because it uses less harsh chemicals, or it almost doesn't use. It's easy to control. No harmful byproducts are released to the market, and enzymes are definitely the answer to the burning issues of the world, be with plastic degradation, municipal waste management, wastewater treatment in STPs, and pharmaceuticals. They are realizing the potential of it. They're rushing towards the usage of enzymes. Even for diagnosis, of late, like since 2022, people are realizing the use of enzymes and the need to engineer them. Any enzyme, for that matter, can be engineered, and there are few exceptions. Let's not talk about it, for the industrial application. And why shouldn't we be the pioneers in that? And I would like to end my talk with, very famous Subhashita in Sanskrit, which says, "Amantram aksharam nasti, nasti moolam anaushadham, ayogya purusho nasti, yojakas tatra durlabha." There is not a single word that cannot become a mantra or that cannot become a sacred chant. There is no plant under the sun which cannot become a medicinal plant. And there is no person in the world who is useless. But what is lacking really is an enabler. So that's what we call Ayogya Purusho Nasti Yojakas Tatra Durlabaha. So it would be really happy to see if somebody gets inspired by this, and then if they start looking into this protein chemistry aspect, or the enzymology or enzyme engineering part of it. I would be really delighted to hear such